What you're seeing is very clever organizational skills. And thank you to Declan and Tom and everyone else at TMRN for the invitation. You put the sample size talk at the end for good reason, but you don't give the statistician the last word of the day either. So there's a fine balance there. So I have a secret title to this talk, and it's this one, Sample Size Theater, what it is, how it hurts us, and striving to do better. For those of you who came for a talk about principles of sample size evaluation, we are going to cover those principles, I assure you, so please don't be alarmed. We just might tack on a few extra thoughts as we move towards the end. Just to give a bit of context for what I do, I've been in this role as the principal statistician for the clinical research facility down at UCC now for 8 years. And so where Erica very kindly or wisely ends with consult the statistician. Well, if you are an investigator, if you're engaged in patient-focused research in Cork, and you don't have piles and piles of money laying around to spend, then you might find yourself to me, and I'd be that statistician that would talk you through your study and try to help you the best that I possibly can. And let's just set a big context here for when we talk about sample size in general, and this is, I was really pleased to hear over and over again. I'll say messages that have been already shared throughout the course, so I'm kind of really happy about that. But it also speaks to how we have so many people working in clinical trials, and we all wind up in the same place, facing the same challenges and all the rest of it. But when we're thinking about a study in a clinical trial, we want to make sure that trial is fit for purpose. It is designed to meet the aims that we have set out to meet with that study. And this is true of all experimental designs, but of course it's especially true for clinical trials because we are doing studies with and for patients. And even the most, supposedly the most mild clinical trial you could think of, there will always be inconvenience for patients. And these aren't college students who are trying to get extra credit in your class. Like, these are people who are not well and seeking care. And any inconvenience to those patients is a huge issue. And we need to make sure that if we're going to inconvenience patients, if we're going to expose them to as of yet unknown risks, that is a part of clinical trials. We are doing things that we don't know the results of. We're asking them to take on a risk. And we're raising hopes. And we're interfering in these people's lives in hopefully a very positive way. And in their families and all the rest of it. But if the design is not there, if we wind up with a clinical trial that can't be used to learn something valuable about care, then we have wasted all of that. And so that is the seriousness that I see. My part of this, and obviously we've seen other talks, all of this has to be together. Now, there's also the very practical element of sample size evaluation, which is that we have to also plan this very large, complicated thing. And has a lot of moving parts, as we've seen, laid out by people who know a lot more about that than I do. But obviously, the number of patients enrolled in the trial is going to have a very significant impact on how your budget is planned, your logistics, and all the rest of it. And then I'll add one more bit of context again for my world, is that, I sit on the NREC, I see commercially sponsored trial protocols through there, but they don't need me, right? So I don't really do any work with any commercially sponsored studies, because they have their own. They have great CROs. They have their own internal teams of rooms full of statisticians. In Cork, it's me, it's Brendan, it's Christine. That's us, right? So it's this. I'm speaking from a context of wanting to see more investigator-led trials, but knowing that the research funding environment for those trials, which we've heard about, is a very, very challenging one indeed. Okay. So let's talk about sample size a bit. And what I'm going to use here, and this isn't picking on the HRB at all, who pay my salary, that's my conflict of interest, them in University College Cork, is how does this advice come? So you decide you want to study, you're looking for an investigator-led study, you need to go and ask for money. And so this is advice from the last DIFA, so the Definitive Intervention Feasibility Awards, and then currently the APRO right now. I would ask if anybody's applying for an APRO, but I don't want you to think I'm spying on you, but this is a due date for next week. And you will see this word power, right? This maybe mystical word power. That is a technical statistical term, and I'm going to talk about power today. Now, this idea of power, I want to say something kind of off to the side about statistics. I'm not a statistician by training. I'm an epidemiologist, a biological anthropologist. I became an applied statistician just through, I don't know, a desire for punishment and all the rest of it. And then got involved in clinical trials and the rest of it. And what I want to say about statistics in general is it is usually conveyed to people as though it is mathematical. It is really much more philosophical. Okay, statistics, more than anything in my opinion, is about thought exercises that help us stop from fooling ourselves with data, right? And power, this technical word power, reflects a particular school of thought in statistics that I think is very valuable, in particular for clinical trials, but it's not the only way of thinking about things, right? So I just want to kind of leave the door open when we come back to this at the end. But you see this word power, and this is what I'm going to talk about today. And the first little hint of sample size theater is that everybody thinks sample size evaluation is about power, and nothing could be further from the truth. This is an idea that, in my opinion, is very well aligned to the idea of a randomized experiment, including clinical trials, but it's not the only way to think about it. But this is the thing that investigators come to me, and I'm tempted to pull out my phone and I show you the emails from the last 2 weeks about the appros coming through. Darren, we need help with our sample size. What's the power for our study? And so that's what I'm going to focus on here. Now this is a cruel, cruel slide. I didn't think it would be this cruel when I put it in, but we're still waiting for spring, I think. But we're getting there, I'm told. I'm 10 years in Ireland, and this is the first year where I'm really, really ready for the next bit. It's been fine up to this point. When you hear the word power, what I want you to do is I want you to think of a metal detector, right? Your trial, as soon as someone says, I want to know the power, I want you to think my trial is a metal detector. And you are on this beach, God bless, and you are looking for treasure. That's how we're going to think about it, right? And power in that context, it's just the probability that you'll detect the thing that you set off to detect. And our job as trialists, as clinical trial designers, the team involved in doing that, is to build the detector that's going to detect what we want to detect with a very high probability. We want a high-powered detector, all right? It's not really any more complicated than that. So let's think then about what some principles might be, things we have to think about if we're designing our detector. And the first principle is that big effects are much easier to find than small ones. Big things buried underneath the sand are much easier to find than wedding rings and earrings, right? And an important key element here is, given the detector in your hand, given the clinical trial you've already designed, it doesn't have one power. It has an infinite number of powers, each to detect an infinite number of things you could possibly detect, right? So one trial design is going to be high-powered for big effects and very low power for small effects. And that is a feature of the design itself. Studies don't have a power, and I think that's an important thing that's sometimes missed. Another thing we have to consider is noise, right? So if there is noise in what we're trying to detect, then obviously it's harder to make sense of what it is that we're detecting. And a high noise situation is going to require consideration in a different instrument than in a low noise situation. In the context of a clinical trial, what are we detecting? We're trying to detect and estimate causal effects of treatments. Those are typically between group comparisons of things. The things we're comparing tend to be averages or some summaries of outcomes in those groups, two groups in a two-arm trial. We know that those summaries are going to be affected by the things they're summarizing, your blood pressure, your height, your quality of life, and that some measures are noisier than others. Some are more naturally variable, some have more measurement error. And so you have to consider how noisy the thing is you're trying to measure when you're building your detector. And then when we think about it, and this could be an oversimplification, and maybe we'll have some questions about that later, but if we're thinking about the trial as a detector, then we're also saying that we're going to act as if. In other words, we're going to get to the end of our trial, and depending on what we find, we're going to act as if this treatment was useful, efficacious, or not. Now, the reality is much, much more complicated than that, isn't it? right? We have multiple studies. We need to be synthesizing evidence. We have, health technology assessment to make decisions around spending, all the rest of it. But again, statistics is about mental experiments to help you keep from fooling yourself. And so when we're talking about detectors, this brings in everybody's favorite, the false negatives and the false positives. So we have a signal from a detector, and then there's some demarcation point where we're going to act as if the thing we're trying to detect was detected, and another one where we're going to act as if we didn't detect the thing. Now, what's really, most people have come across this kind of thinking. It exists in tests, sensitivities and specificities and those kinds of things. But what we really have to understand is that these errors have different costs, right? So if you're walking around and you're beachcombing with your metal detector and it goes off constantly, right? It's just a constant stream of false positives. That's obviously going to be a huge waste of your time, in trying to do something, in trying to find something. You're going to waste all your time looking, down, dead ends. False negatives are a different thing. You might miss the little piece of treasure that's there, the most valuable thing there could have been on the beach. Typically, in clinical trial setting, we tend to preference a stricter restriction on false positive findings. Now, that doesn't have to be one way. We do need to be more thoughtful about this, but culturally, for lack of a better word, we tend to think to ourselves, well, a false positive, once something's on the market, so to speak, very hard to undo that, right? Very hard to then get equipoise has been disturbed. You're trying to say, well, we could study this more, but we actually have pretty good evidence. False positives are very hard to undo. But false negatives aren't nothing. False negatives, which you usually accept a higher false negative rate in a trial, that's a missed opportunity for an efficacious treatment, right? So this is serious business as well. And an important thing about this is we can always, given the tool, given the trial we've designed, we can always trade off false negatives and false positives. In other words, the trial, the detector produces a signal. I have some cutoff somewhere that says I'm going to act as if it works. If it crosses this threshold, I'm going to act as if it doesn't. If it doesn't, I can always change that threshold, and I can always create a tool that will give me 0 false negatives. Right? It goes off all the time. I can give me a tool that gives me 0 false positives. It never goes off. And so we can always trade that off in the design. And then finally, we can just improve our detector, right? We can just, oomph it up. And the way to do that, overwhelmingly, is to increase the sample size of the study. All right. Now, we could use a less variable outcome, and I'll come back to this in just a second. But this is the typical thing. The power comes from the sample size. And what's tricky, though, and where we're now way different from detectors, is there are different costs for us increasing our power by increasing the sample size. And so this quote is great. This is from Steven Sen's book. If you're interested in trial design, he's written a wonderful book, Statistical Issues in Drug Development. And this quote is in there. It's not his quote, but it's where I stole it from, his book. And it's anybody can build a bridge, but it takes an engineer to build a bridge that just doesn't collapse. And what he means by that is, you're trying to design a thing that detects an effect in a trial, but we don't want to over-detect it, because the only way to over-detect it is to have more patients than we really need, and patients are a precious, precious resource. Ethically, morally, logistically, financially, right? And so to stretch I'm going to risk another analogy here, is we're trying to land the plane with a trial. But we're trying to land it on a very short runway, right? We're trying to have enough power, a good enough design to detect what we need to detect, but we can't overshoot the mark either, right? Because patients just aren't out there easy enough. This is the key challenge of trials, right? So that, from a design element, brings in a lot of sophistication and nuance and the rest of it that has to go into how we properly design a trial. We can't just build a bridge by taking a bulldozer and ramming a bunch of dirt up into a hill over a ravine and go over it. We've got to build a thing with a very constrained number of resources. Now, I won't linger here, but this is a thing where we need to consider all the stuff we've talked about. And in a 20-25 minute talk, Obviously, I can't give a lesson on how to do a sample size calculation. We're just talking about principles. But this is, just so you know, this is how I think of it. And if this is an image, a diagram that you're familiar with, then you're in a pretty good spot. And if you're trying to design trials and you're not familiar with these things I have an outcome I'm measuring, some statistic I'm calculating from the data in the trial that reflects this causal effect of treatment. I have some null value, 0 for a randomized trial. I have a distribution that says the expected distribution under sampling variability. In other words, if I imagined a trial that didn't do anything, that you had an intervention where nothing happened to anybody, and this number I'm calculating, the treatment effect, it's just a function of who I happen to randomize into what arm. I take everybody in this group, I measure your blood pressure, I randomly allocate you, I compare the mean in both groups, I do it again, re-randomize you. I'm not going to get the same answer for the mean. There's a distribution. We need to know what that is. And then we imagine that distribution centered on different effect sizes. And all of this stuff that I'm talking about has to come together. And if you're trying to design trials, this is the kind of stuff you have to learn and engage with. And I'll say this as a starting point. This is a particularly wonderful interaction app that you hop online. And you can play with these numbers. The point is that all of these things I'm talking about, the sample size, the outcome variability, the false positive, false negatives, the effect size I'm looking at, all of these things in a design trade off against each other. Right? So that's the conversation that has to be had. Ellen, how am I doing?
You have 8 minutes.
That's good. I got time to get fired up. That's good. A simpler way to think about this is just a simple signal to noise ratio when we're thinking about a trial in our detector. And the signal is the effect size. It's the thing we're trying to detect, the causal effect of the intervention as it's estimated by our trial. The noise is in itself a ratio, again, of the outcome variability. How noisy, how variable is the outcome you're interested in. All causal effects are stated in some kind of outcome scale. This is affecting my blood pressure. Is it affecting my quality of life? And then the sample size. So the sample size goes up. The denominator comes down, the noise comes down, your signal-to-noise ratio gets better, right? Just basic stuff. It's really not more complicated than that. Now, this is where we get into me ranting for a minute about sample size theater. In a perfect world, you would follow Erica's advice, you would consult the statistician, you would think about all of these things that it requires. 1, an effect size. And a good way of thinking about effect sizes is, again, I'm going to steal from Stephen Sin. You're trying to think about the smallest effect size that's clinically important, an effect you'd like to not miss. If we go back and think about the false negative rates in the trials, we usually accept a power of maybe 80% or 90%. 100 minus that is this false negative rate. We usually accept 20%. False negatives. If we were to imagine rerunning our trial an infinite number of times, a thought experiment, the way we design trials would often accept that 10% of those times in these infinite number of hypothetical universes where we're running these trials, I'm telling you, it's all made up, it's not math, that we would accept 20% of the time not finding an effect that we set out to detect. And that's a real cost. So you really need to think carefully. What's the smallest thing that we'd like to not miss? And then, if you power your study for that, anything bigger than that, you will automatically be well powered for. So it kind of reduces this into a somewhat simpler problem. You would carefully consider the costs of these errors. You would carefully consider a false positive versus a false negative, and how can we achieve the balance that we need. You would think about the variability of your outcome. You would come to the statistician, you'd say, we're going to do blood pressure. I know in this particular group of people at this particular time of year, this is the variability. I looked up, I did all the research, and I've got it. And with all of that information, we would work together, and we would come up with a good guess. Calculation's almost not even the right word. There's a calculation on top of this that you do need the statistician for, but that's the easy part. These are all clinical questions. I don't know any of this. I can't. So that's for you to do. Then you come to the statistician, and if you can do all this, it's not quite cranking a handle. There's a lot of other nuances and everything else, but it's not that far off. Now, what happens in reality? Ugh. Yeah, here we go. You email 2 days before the grant is due. You tell them, well, we used this many patients the last time. And I don't mean you. And I'm not trying to put this on you, by the way, because we're going to come back to this. This is a systemic structural issue that we all suffer from, not a statistician wagging their finger at you, the investigator. You tell them how many patients were in the previous study. You follow some convention on what the alpha is, the type 1 error, what the power is. You make some wild guess about outcome variability. That's the one I do find the most puzzling. Why people don't know this more often. And you pray that the resulting thing that comes out of this sounds like something would fund. It sounds reasonable. And what happens is, in this little performance, is that we write it up and we pretend that it's very, very scientific and that we have estimated the samples, we have calculated the sample size for this study. We send it to a grant panel, to reviewers. Maybe there's a statistician or two. Maybe there's an experienced clinical trialist who understands, this stuff. And that's how things move forward, right? It is theater. And this is where I get preachy. We've got to stop conducting this theater because it comes back to that stuff about the patients. We are wasting a precious resource. We are generating massive amounts of research waste. Every paper that goes out and looks at the quality of the scientific literature in biomedicine tells the same story. We are wasting. And this is part of the reason for that. And I'll come back to this airplane thing. So what happens in practice here, there's 2 more slides, is the first problem is that, particularly for the investigator-led stuff, we're being asked to do all this complicated stuff before we have the grant, right? Now, I'm funded piece of infrastructure. The HRB has done a very smart thing in recognizing the need for infrastructure. So I am core funded by the college, by the university, and by the HRB to sit in my little cubbyhole and wait for investigators to come and talk to me, and that's what I happily do, right? But this isn't to complain. I love my job. There's only one of me, right? There's one of me in Cork, right? The scale we're talking about is just totally different. And it's not just trials for me. I try to cover all the patient-focused research. So I have speech-language pathologists, I have nurses coming over, I have physiotherapists. And so it's very hard to pretend like I can sit down and have the time for everybody that wants to get a grant to really plan out their study, right? But again, we're going to do the theater. And if we get the grant, now we're going to sit down and we're going to actually plan the study. It's not that we didn't plan it. Who's here? I don't even know who's here. This isn't recorded, is it? We do give it a lot of thought at the application stage, but obviously, if the resource isn't there, there's only so much you can do. And so we need, it's not me, it's not just statisticians, all the stuff everybody's talked about, we need all of that. And we need time for investigators, because I wake up, I work very hard, but I work in my pajamas. I go to Starbucks and work, right? I don't see patients. That's hard. And that's what we're asking those people in very little time to do all this work with me. It's ridiculous. And then the other thing to kind of land where Alistair landed, talking about adaptive trials, response adaptive trials, platforms, and the rest of it, that's like the future. That's like unicorn stuff, right? What we actually do is we take a design at the beginning and we put a mannequin at the controls and we launch the plane. And yeah, some things change during the course of the trial. We make amendments, we make tweaks and whatnot. But if we really wanted to get away from how we do things, then many, many more trials would be completely adaptive, right? We would learn, we know that all that stuff that went into the trial's design was just our best guess, an educated guess, and we know it's not right, right? So you can calculate the flight plan, you can launch the plane, But you need a pilot to steer when there's the wind and all the rest of it, right? But you need those things that Alistair was talking about. You need infrastructure, you need data, you need statisticians. He talked about Barry Consulting. Those are smart people who do this full time to design those types of trials, right? So we need more in that way. Okay, I will shut up before Ellen starts bullying me. So anyway, thank you very much.